Welcome everybody. I'm Travis Ross, the manager of the Visualization Lab. I think we'll get started. So welcome to the very first of the new Beckman TechTalks. This is a new series of talks that between Scott Robinson, Brad Sutton, and myself, and kind of the inspiration of several other people here in Beckman, we decided would be a good thing to initiate. I don't know if some of you remember years ago we used to have the imaging forums, and we stopped doing that, and there was kind of a void there for a long time. We weren't quite sure how to fill that void, but we knew we needed something to talk about some of the really neat, cool technologies that we have in the facilities here in the building. So today is the first of what we call Tech Talks on Tuesday, and these are always going to be from now on until the end of the spring semester, the second Tuesday of the month, given anyone's going to be around, because I think January we might not be having one, because I don't think very many people will be here on that Tuesday. And today we're going to focus on the visualization lab, and then specifically we're going to talk about one piece of equipment that we have that's fairly new, and it's our high-speed camera. And for that part of the talk, I'll be introducing Josh Gibson, who's up here in the front. And the way this will work every month is that we'll cycle through. One month will be the Viz Lab, the next month will be Microscopy Suite, the next month will be BIC, and then the next month we'll come back to the Viz Lab, and then Microscopy Suite and BIC. And we're going to cycle through these three facilities. That way, eventually, we hope in the next couple of years, if we continue to do these talks, we'll cover just about every single piece of technology, software, hardware, pipeline that we have, so that those of you who are around Beckman community who know about us, you know more specifically what we might be able to do to help you. Because that's really our goal here, is to facilitate your research. And it's always been my dream to have everything under the sun as far as visualization, but we can't make that happen, and we have a small staff, and we do the best we can, obviously. But what I want to see happen out of this is for you to get some ideas in your mind about what we might be able to do to help you down the road. Maybe what you see today won't help you immediately, but you might remember something you see here, or you might see something next month or in a few months that you think that might be helpful, and then the next time you have that need, you just come see us. And one of the things I always like to say about the Viz Lab is that we're kind of a boutique facility. Yes, we have a dedicated or a specific number of computers and seats of software and time of the day, but we're really available to sit down and talk with anyone who has a real specific need. And we're able to tweak our hardware and our software and install new things, and we have the flexibility within reason to purchase new things as needed to make your work easier. And that's my goal. I want to see everyone here succeeding. That kind of gets me excited. So with that, I'm going to give a small presentation of the basic overview of the Viz Lab. And we have a lot of things that I'm not really going to be able to spend a lot of time talking about, but I'm kind of give an overview of what we have. So, as the title implies, we're the Visualization Lab, and that really is pretty broad. I mean, we cover everything from graphic software to computational analysis, high-speed video, 3D scanning, 3D CAD, and we help people apply those technologies. We don't do any development of software. Maybe we did a few years ago. Actually, before I was here, there was some coders, programmers who were writing new software. And we typically just buy things off the shelf, kind of plug them together, and put together a pipeline. I mean, that's what I think we're really good at doing, is helping select software and hardware that will make things easier for you, and then tweaking those as needed. So that's kind of a broad overview of what we do. This is our staff. It does change from time to time. And of course, that's me on the left there with a beard that went away a couple weeks ago when I went on vacation. And then Alex Jerez, who's been around a lot longer than me. He's one of our 3D graphics guys, and if you've ever had a journal cover designed by us, you've probably talked with him. Great experience in 3D graphics industry. And then Jose next to him. Jose has worked with Alex. I've worked with Jose for several years now. Another really good artist. The guy's amazing with Photoshop. really good with just pencil and paper. If you need a concept, we sit down with you. What we'll do is within a half an hour, we can come up with design concepts really on the fly. We can change those quickly, and that's what these guys are really good at doing. So if there's a journal cover, maybe you need an image for a presentation, that is something really special and specific that we've always provided to a lot of people here in Beckman. And then Peyton on the end, he's a younger graphics guy. He's been around here for a few years as well, and he's an excellent 3D graphics specialist. And then on the bottom we have the newest addition is Ariel, who's sitting right here in the front. Ariel joined us after graduating from the business school last year. And if you haven't noticed, her picture is actually on the sidewalk leading up to Beckman from the parking garage, if you haven't seen that, with her glove, one of her racing gloves in hand. She's on the Illinois wheelchair racing team. And then Kendra, our wonderful Kendra, who helps, where is she? Everything from purchasing to making sure you have enough food in the back, to renewing contracts for us with the help of our wonderful business office. So that's our staff. So what do we have in terms of software? We have image analysis and quantification software, and I kind of lump those together because I really need a way to kind of organize them. But we have stuff like Amira, which allows you to take MRI and CT image stack data and turn those into 3D volumes, like on the very top image there. Aviso is another one that we just acquired, which is kind of a cousin to Amira. It does the same thing, but it's more specifically for material scientists. And then Image Pro Plus, which is kind of a lesser known software. It's excellent for taking the 2D slices and creating some kind of quantification, analysis, or measurement just within the 2D image itself. And then 3D CAD simulation analysis software. Console is the big one for us, really. I'd say most of our users in the Viz Lab who are sitting on a machine, they're using Console. And I think we're up to, I've kind of lost track, honestly, of how many licenses we have. We just keep adding more and more licenses as the need arises. So when enough people start complaining we're out of licenses on our server, we'll just add more. Abacus and Ansys, two other really popular softwares for us. And I will say I'm not an expert in either of those three softwares. I'm just really good at facilitating them and making sure they work. And honestly, if you come to me, I'm going to direct you to, if you come to me and you've never used them, I'm going to direct you to others who have and online resources for learning those things. Because they're so specific to each individual's research work, honestly. I know how to launch software, get in the interface, and move around, but really everything in those softwares is going to need to be pretty much tailored to what you do. And then, of course, we have other 3D CAD design softwares like SolidWorks and Inventor and AutoCAD. 3D modeling and what we call visual effects software is what we use to create any kind of lifelike looking synthetic image that's not photographed. If you need something that is imaginative and you need it rendered, there's not a way to photograph it, we will recreate that in 3D. We can take data sets, 3D data sets from just about anywhere, and we'll turn that into an image. We'll actually render it, and we've done that many, many times for our journal covers. And we use a lot of a large combination of 3D design software, graphics editing software, Photoshop, Illustrator, and then those same softwares are also available to you. So if you want to come in and use those softwares, they're there. They're there for everyone. And then we still have video, audio, and multimedia production software, even though we no longer have our sound booth. So if you haven't been downstairs, the Viz Lab is actually directly beneath us, 4602. We used to actually have a full production video and audio setup down there, but as times have changed, we gave that whole setup to the main to the undergrad library in their media commons, so you can still use that equipment. But we kept a lot of the software that people might need to use to edit audio and video and create nice productions for their presentations. So you can still use that there too. I want to mention briefly our computer workstations. They're not a typical computer lab type computer. They're all very high end. We typically have been buying HP Z-class computers according to what ITS has helped us spec out. And by the way, these guys are wonderful. Up here on the 5th floor, they get really tired of hearing from me probably every day about things that break, but they're great at helping us fix the lab and keep the computers and our network running continuously. I mean, they are, without them, we would not be working as efficiently as we do. But these machines are up to 32 cores with up to 512 gigs of memory. That specific configuration is on a remote machine that isn't sitting in the lab. The ones that are in the lab are all 16 cores and up to 256 gigs of memory. NVIDIA GPUs, large monitors, and I'm going to start getting the curved displays in the next set of computers because they are so amazing. I love them. I have one on my computer. So you need to reserve our computer, sit down, and I didn't add this to the slide, but it's an hourly rate. It's how we charge our, that's how we keep our lab open, is by charging an hourly rate, and that allows us to maintain our software, not purchase our software necessarily. All of our purchases, our hardware, our software, is all purchased through the generous Beckman Foundation gift money that we receive every year. We get that money, we prioritize what purchases we think we need, and then we make a purchase from that money. Then the money that you all pay to use our lab, that goes to maintain everything. So if the hard drive breaks or a software maintenance contract renewal comes, the money that money that you pay towards the Viz Lab covers that cost. And then a little bit about some other specific things. Our high-speed video camera, this is it. This is our newest addition to what we have. It's the Photron FastCam SAZ. That's just a specific model because it had the most amount of memory and everything that we needed to be flexible. And that's always what we try to do, is be extremely flexible in what we buy. I try not to buy things that are only intended for one narrow purpose. I want to make sure it's useful for as many people as possible. And we purchase things based off of advice and input that we get from our users. And that's the reason we want to hear from everyone is what is it you need to be successful. And then this is what Josh is going to talk about. I will mention, I don't have a slide on this, but we have 2 other high-speed cameras, and one of them is extremely portable. It comes in a roll-behind case, and you can take that with you to your lab. It comes with a laptop, tripod, and everything, and it's really easy to use. I can train people how to use these cameras in 30 minutes, and then you can start capturing video for your research as well. Downstairs, sorry, I forgot to mention the high-speed camera, the macro photography, and then the 3D scanner. They're in a downstairs room in the basement in room B650G. That room was sort of transitioned over to us, I don't know, maybe 2 years ago, and we refurbished it and redesigned it for our purpose. It's got a very high ceiling. We have a lighting track that hangs from the ceiling. We move our lights around, and it acts almost like a photo studio / video studio as well. It's very, very versatile. Everything's on wheels. Tall camera stands that can support the heavy weight of these cameras. And then within that room is where we have the high-speed camera, and then this one as well. And then the macro, so the macro setup is a little bit more of a dedicated camera setup, and many of you have used that, but it has 50 megapixel DSLR, a really wide array of lenses for just about anything from point and shoot, outdoor camera photography, to microscopic high-resolution video, if need be, which we've done everything in between. And there are a few images right there on the screen that we've captured within the past few months or years. And then something that gets used quite extensively is what's called the Z-stacking device. That is an automated system that controls kind of the camera Z direction and then automatically takes a series of pictures and then stacks those all together into a nice in-focus picture that is definitely impossible to capture when you're using a microscopic lens on these cameras. I've gotten pretty good at using that, and there's almost virtually no instruction how to use these things. It's just been through experimentation and just a lot of tinkering to make them work, because the lenses are quite difficult to use, a microscopic lens on a DSLR camera, and they're all kind of custom-built lenses. And so come and see us if you have a small object that you need a nice, good, crisp, clear image of for publication. We'd be happy to help you with that. And then 3D scanning. Our 3D scanner is now going to be mostly in Ariel's domain, because she's the one who's been using it so much for her work before she graduated. But I want to start sending her as much of this work as we can. But the scanner it's a Steinbichler, which is now owned by Carl Zeiss, I believe. Sorry, I can't remember. But it can be used for measurement, analysis, capturing a shape that can then be redesigned. Design, Dina? See, I mentioned design.
Woo!
Okay. Design is a big thing. We partner with Dina and the art department and so maybe we'll do a presentation on that someday. And rapid prototyping. So such as 3D scanning a statue that was in Peter Schiffer's office for years, and then when he left, we created a 3D printed replica of it. So that's what that is really good at doing. It's portable also. It doesn't have to just be used downstairs. It can be taken even outside. That's actually on the right a picture of the Alma when it was sitting in a warehouse in Forest Park, Illinois, when it was being rehabbed, and we took our scanner there and spent 4 days 3D scanning her. And then again, about our special projects team. So within this team, we've done everything from simple hand-drawn concepts, just to quickly communicate an idea for someone, to immersive environments. Very, very diverse, very talented team of people that come out of the industry. And that's really important because it takes a lot of years to learn 3D technologies and graphics and visualization tools. And so what they're good at doing is taking professional tools out of the industry and applying them to making images look really good for your research and journal covers. So if you have a journal cover need, come see us. It doesn't cost you anything to just have a consultation. We'll sit down and talk to you guys about what it is you are trying to communicate. And always, I think one of the most important questions we're always going to ask you is, what is it about your research that you want to promote in this one image that you've been, working on for the past so many years? What is that one thing that you need your audience, and who is your audience, and how do you want to communicate that to them? And let us figure out a way to come up with a nice image that will kind of make that come alive. So that's the end of the VizLab overview. I want to mention the Tech Talks that are coming up. We've got the next one on November 14th. Scott will be talking about microscopy suite, new instrumentation, and the future of things that he has in mind for that facility. And then on December 12th, Brad will be talking about Biomedical Imaging Center. And so as part of all these talks, the hope is that we'll be having researchers who have benefited from our labs then talking specifically about that. And so as soon as I'm done, Josh is going to come up. When Josh is finished, I invite anyone to come downstairs to B650G in the studio, and we'll actually do a demonstration and some more, if you want, hands-on with the Highspeed camera. And I'll even extend that even into the next week or two. If you have a project need and you're not sure how this might work for you, just come and see me. I'm either in the Viz Lab right below us, or in my office, or somewhere in between there in the basement, or running around the building. You can catch me through email too pretty easily, which is right there. So yeah, right after Josh is done, we'll go downstairs. Anyone who's interested, we'll do a demonstration of the Highspeed camera. And then Josh, you can come on up.
I'm an entomology graduate student, and I'm a biologist, and I work on a group of animals that have what are called power amplification mechanisms. So these are mechanisms that, through the use of a series of springs, levers, and latches, allow for animals to store and transfer mechanical and elastic energy within their bodies. You can kind of think of these mechanisms as being similar to a slingshot. So if you have a slingshot and you want to throw a ball, you put the ball in the basket of the slingshot and then pull back. And that stores elastic energy within the rubber bands on either side of the slingshot. And then when you're ready to release the ball, you let go of your fingers, which are latching the slingshot open, and the ball goes flying, because all that energy is released at once. Animals that have power amplification mechanisms do something very similar, only instead of launching a ball, they're moving their limbs or mandibles. And instead of using a rubber band to store energy, they're using some aspect of their cuticle or muscle tendons and muscles themselves. So these mechanisms are relatively common across animals, but especially in arthropods, because arthropods tend to be relatively small-bodied, and because of this have constraints on how fast they can move because they don't have much time to accelerate their limbs and have smaller muscles. But using these mechanisms, they're able to move their limbs faster than possible through muscle contraction alone, and actually using these mechanisms are able to generate some of the fastest movements that we know of within animals. So often they use these mechanisms as a means to escape prey, which is the case in things like this grasshopper, but also springtails and plant hoppers, or as a means to capture fast-moving or elusive prey if they're predators, which is the case in things like this mantis shrimp, but also trapjaw spiders and trapjaw ants. And understanding how these mechanisms work is important because, especially in a biological inspired design perspective, because if engineers are trying to design maybe a small robot that can jump very far, they can look upon things like grasshoppers or other small power amplifying jumping insects and then replicate those designs in a robotic system. So this is a robot that was designed after inspired by the jump of a grasshopper. And then also the things that act as the springs, so the cuticle and tendons, sometimes have really interesting elastic properties to them that potentially we can replicate in other systems. So it's important for that aspect. So I work on a group of power amplifying animals called trapjaw ants. So these are, is this the laser pointer? Yeah. So these are a group of ants that have really elongated mandibles. They're able to be locked open at a wide gape. And this allows the ant to store energy elastically within their head capsule as they contract enlarged mandible closer muscles. And then when they're kind of hard to see, but they're long mechanosensory hairs that are called trigger hairs. When those are stimulated by prey or other stimuli, the mandibles are released and slam shut at astounding speeds, sometimes over 100 miles an hour, and the ant can generate forces over 300 times the weight of their body. So it's an incredibly powerful mechanism for these ants. And one of the cool things about trapjaw ant mechanisms is that not one group of ants have evolved them, but actually at least 4 groups of ants. And I'll talk later about a project that's shown that it's actually evolved more than 4 times, but at least 4 groups of ants have evolved this mechanism in 3 different subfamilies. So it's a really great system to look at conversion evolution of power amplification mechanisms, comparative biomechanics of these mechanisms, look at things like how power amplification mechanisms influence diversification rates, and things like that. So I work mainly on these 2 groups of trapjaw ants. These are the Daceton genus group and then the genus Strimigenes. And so when I was invited to talk to you guys, when I was initially invited to talk to you guys, I was told that I should probably talk about how the Viz Lab has allowed me to advance my research. And so I'm going to be talking about 2 different instances where advances in technology at the Viz Lab have allowed me to visualize things that I would not normally be able to visualize. And the first is looking at strike kinematics in the genus Strimigenes, and the second is looking at latch release in Daceton. So for Strimigenes, these are miniature trapjaw ants, is their common name. And they get this name because, despite what this picture might lead you to believe, they're really tiny. So they're about 1 millimeter to 2 millimeters long. So this is a picture of a neotropical species that's on my pinky finger. But they're incredibly morphologically diverse in species. So there's a wide amount of variation in terms of the mandible shape and size, as well as head capsule shape and size. And there's around 850 described species worldwide. So it's a great group to look at not only comparative biomechanics of trapjaw mechanisms and evolution of trapjaw mechanisms, but also the relationship between form and function and performance in these guys. So when I first got here in August of my first field season in August of 2015, I decided to go down and do some preliminary studies on these guys. So I went down to Georgia to collect the only native North American Strimigenes that has a trapjaw mechanism, which is Strimigenes louisianae. So I went down to Georgia, collected the ants, and then went back to Illinois, brought them to the Viz Lab, and tried filming them with the setup that our lab used a lot at the time, which consists of this Phantom 9.1 high-speed camera attached to a stereo microscope in the microscopy suite, and we would put the ants down here, restrain them by their head, and then backlight them by aiming lights at a mirror which shoots the light up. Because when you're filming at really high frame rates with these cameras, your light becomes an issue, and so the only way you can really visualize these strikes is by backlighting them. And so we filmed at what was essentially our maximum frame rate that we could get excuse me, using this setup, which is around 120,000 frames per second. And this is the video that we got. As you can see, or rather not see, the strike happens within one frame. So we're not actually able to visualize what's going on here. We know that they're really fast, but it's not really going to give us meaningful data using this setup. So I had to take my ants and fly to North Carolina about a month later and visit a collaborator's lab at Duke University, Sheila Patek. And she had a better setup. So Travis mentioned an SAZ earlier. At the time, Beckman didn't have one, but her lab did. So we mounted it to a microscope, sort of imitating our previous setup, and we were able to film at around 1 million frames per second using this setup. And we were able to finally get good data, and this is an example video that we were able to take. So this structure right here is the labrum, which is what acts as the latch in this group, and you can see it drop down and the mandibles are released. And with this species, the mandibles close in less than 0.018 milliseconds, which is about 15,000 times faster than the blink of a human eye. They can move their mandibles at around 55 meters per second on average. Some individuals get up to around 70 meters per second. And they accelerate their mandibles at around 15 million meters per second squared. So these are insanely fast ants, and they're insanely fast even for trapjaw ants. So they reach similar velocities to other trapjaw ants, but their strike durations and accelerations are an order of magnitude faster than other trapjaw ant groups that we've worked with. But most of the diversity of these guys is concentrated in the tropics, and so if I wanted to go down and get a wide sample of a species to study, it wouldn't really be feasible for me to fly to someplace like Brazil, bring the ants back to Illinois, wait a month or two, and then fly to North Carolina to film these guys using Sheila's setup. The other thing about these ants is that they're very finicky and kind of hard to keep alive in the lab, so there would be no guarantee that I would even be able to keep them alive long enough to get to Duke again. So fortunately, about a year later, the Viz Lab acquired their own SAZ, and so using this setup here, we were able to film the ants at Beckman, and using this, we were able to add an additional 14 species to our data set. And these species are from around the world, places like Mozambique, French Guiana, and Peru. And this is just, I don't want to focus too much on the data that we got, but you can kind of see they're all reaching a pretty similar strike velocities with the exception of this group over here, which are ridiculously slower. And so we actually found that these guys are not using a power amplification mechanism. And if you look at the relationship, the evolutionary relationships between the ants that are using these mechanisms and the ants that aren't, you see that the power amplifying trapjaw mandible forms have evolved from non-power amplifying groups at least 4 and possibly 5 times in this genus. So it's a great example of convergent evolution of trapjaw mechanisms even within a single genus. So it's really cool. So I'm going to switch gears and talk about another sort of very preliminary project that we're working on, and that's looking at latch release in trapjaw ants. So in trapjaw ants, or in myrmecine trapjaw ants like Stromygenes and the dacetines, the labrum is what acts as the latch, so it's sort of like akin to the upper lip in humans. And when this is raised up, when the mandibles are open, the mandibles are locked in place, so they can't close. And then when it's pulled down, the mandibles are released. And when it comes to studying power amplification mechanisms, most research focuses on just the performance output of the mechanisms. So like looking at, in this case, how the mandibles would move. But not a lot has been done on latch release, and that's sort of because in most groups, the latch is usually internal or very hard to visualize, and it also is thought to move considerably faster than the mandibles themselves. But we decided to look at this in trapjaw ants because the labrum is quite visible if you look at it from the right direction, and so it's easy to visualize what's going on. So we used a different species of trapjaw ants in this group, and for this study, we used the species Daceton armigerum. And this was just to look at the kinematics of the strikes. So this is an example video. But using this mechanism, this setup, we're not actually able to really visualize latch release because we have to backlight. And if we want to get a front-on view of the latch and actually see what's going on, we wouldn't be able to do that unless we could use direct lighting. So this setup would not work for this. So we later went back and grabbed more ants after the Viz Lab acquired the SAZ. And this is an example video of the same setup. This one's filmed around 100,000 frames per second. Oh, the last one was filmed at 80,000. And you can see we're able to visualize a lot more of what's going on with the mandibles. And you can actually see the latch released. And if we repeat this looking head on, we're able to actually see what's going on. And so I'll show this video, and there's an initial phase that's gonna play back, or was filmed at 300 frames per second. And that's gonna show the loading, and the mandibles will actually slide along the base of the latch, so there's some dynamic movement going on there. And then when the latch is released, it switches to 30,000 frames per second. And it's not just an instantaneous process. There's actually some friction going on, and it takes a while for it to be released. So I'll show the video. It'll play back again. There we go. Those are forceps in the way. So you can see the way mandibles slide up along the latch. This is the latch. And then the latch is pulled down slowly. So if you look at the timing of that, the loading takes around 85 milliseconds. The latch release takes around 0.9 milliseconds. And that's considerably slower than the strike duration, which is around 0.13 milliseconds. So this is still sort of preliminary. We want to do more stuff with this, but it's been pretty cool so far, and I'm excited about how it's turning out. In addition to using high-speed camera equipment in the Viz Lab, I also use a lot of their software at the computer lab. So with the microscopy suite, I do a lot of CT scans to look at the internal morphology of these mechanisms. So this is an example of Stribes louisianus head capsule, and I use a mirror to label those scans. And we're also looking into doing other projects outside of trapjaw ants, looking at polymorphic species of ants and how mandible shape influences the stresses that their mandibles experience when they bite, using FEA models and ANSYS. So just to quickly summarize, my research would not be possible without the advancements in visualization lab technology. I would not really have been feasible for me to continue flying to Duke whenever I needed to get data. So I really appreciate that they've been able to accommodate us and acquire new technologies. And trapjaw ants are insanely fast and insanely cool, and I hope you guys learned something new about them and enjoyed hearing about them. So tons of people that like release itself. So the loading phase is so much slower. Those videos are around 10 gigs because we're saving like 25,000 frames.
Yeah, I'm wondering, do you have anything for really low light imaging? For example, can you capture something that has very low light level and very high speed? For example, looking at a flash of light containing several hundreds, several thousand photons.
Yeah.
Very high speed.
Yeah. I mean, this. I don't remember the ISO numbers off the top of my head, but you wouldn't use any secondary light sources. You would just look directly at your light. Honestly, I would just need to try it and see. Sometimes I can't answer until we put the camera up in front of it and see what happens. Yes, John.
So the purpose of the jamming ant is for catching prey. So the speed it needs, or the crushing power?
It sort of depends on the group. So with Stromygenes, they're actually, as far as we know, all 837 species, or most of the species, are specialists on springtails, which are these power amplifying jumpers. So it's thought that they probably are using the mechanism just to overcome that speed and just be faster and catch the organisms. But the first trap giant I showed, the black one, is Odontomachus, and they are a bit larger and they're more generalists, and they have mandibles that are more shaped like the edges of like a blade or something like that. And so they're basically just going up and cutting things and bludgeoning them to death. And so that's probably they would need more force. And also, some groups of these guys actually can use their mandibles to jump. So Odontomachus, the black ones, do that quite frequently. And so it's thought that they've actually co-opted their mandibles. So it could be that their mandibles are sort of some intermediate form that both optimizes their ability to cut and to jump.
That's right. You can watch these videos of these ants like flipping through the air as they're trying to escape.
Yeah, the Stromygenes can do it as well, but they live in the leaf litter and they probably aren't using it as probably just a byproduct of how much force they're generating relative to their body weight. The Daceton can also kind of do it, but they have really strong grips on things because they live in trees, so they rarely would jump in the wild.
So downstairs, if you're still interested in coming down to the demo, we don't have an ant because ants are a little bit difficult to set up sometimes easily, quickly. They're going to show up.
It's a praying mantis. Yeah, they're a little easier to work with. You have a question? Yeah.
Yeah, are you going to post the talk?
Yeah, thank you for mentioning that. We're going to post these on a new YouTube channel, I believe. That's what we decided to do. We don't have that created yet. That'll be in the future marketing material. So that's the other thing is we want to save these for people who can't be here for future use. Just as in terms of like maybe a little bit of like training material. We'll get very specific into certain technologies and things. People can refer back to those to learn from what they're doing. Questions? Anyone else? Yeah.
What was used to get the video of the same color?
Oh, that's just a regular camera. Yeah, there was a large number of extension tubes that were attached to it, and it was I think a 100mm lens. It wasn't done in the Viz lab; it was done with a former postdoc in our lab's camera.
So you can take any lens and move it further away from the camera and get a little bit of magnification.
Yeah, so I think this was like the camera, and then there was a lens about that long, and then that's how we got it. It was kind of ridiculous.
Yeah.
But
I just love the branding, Dr. Stranger.
yeah.
Sound like a rapper.
You can actually blame Scott for that. So when Bugscope was still actively going on, I don't know what I said, but I posted some joke just in between me and Travis, and he said that I was like a combination between E.O. Wilson and Dr. Strangelove. And then Tanya Josek, another graduate student who was helping out, drew a picture that she sent to me of like Dr. Strangelove with a trapjaw ant head, and I thought that was funny. And I had to come up with a Twitter handle for a class, and that's what I came up with.
So I got a t-shirt.
A T-shirt of it? Okay.
Anyone else? No? Well, thanks for coming, and come back here on the 14th, November 14th, and we'll hear from Scott from my cross